Hello, my name is Liam Wong, and today we're going to be talking about the anatomy and enzymology of fat absorption through the GI tract. So we will begin through a process called catabolism, which is the breakdown of large chemical structures called polymers to monomers so that fat absorption is easier and more efficient. The main polymer we're going to be ingesting through our food and digesting is called triglycerides. This is the most prevalent lipid, and we're going to break it down to monoacylglycerols and fatty acids. Two less common polymers that we'll be ingesting are cholesterol ester and phospholipids. We're going to break down the cholesterol ester to simply cholesterol and phospholipids to glycerol and fatty acids. Now, the process of breaking down polymers to monomers is carried out through enzymatic activity through lipases. We have lingual lipase, gastric lipase, and pancreatic lipase, which we're going to talk about on the next slide, and two other lipases for these specifically for the two less common polymers. So when food enters the oral cavity, our sublingual gland and our parotid gland in the oral cavity will secrete the lingual lipase, which will start the breakdown of these triglycerides and cholesterol esters and phospholipids. And the stomach, the chief cells, will secrete gastric lipase. So we have gastric lipase and lingual lipase secreted in the oral cavity and stomach. And as the food passes into the small intestine, the first part of the small intestine is duodenum. And in the duodenum, the pancreas will secrete pancreatic lipase. This is the biggest contributor of the breakdown of triglycerides to monoacylglycerols and fatty acids. And for those two less common polymers, we have cholesterol ester hydrolase, which will break down that cholesterol ester to cholesterol, and phospholipase A2, PLA2, will break down those phospholipids to glycerol and fatty acids. So, in the duodenum, the first part of the small intestine, we will be secreting not only that pancreatic lipase, but something extremely important called bile. And bile is secreted by the liver and stored in the gallbladder. It will concentrate the bile as well. So secreted in the liver, travel down the hepatic duct, stored in the gallbladder. It'll travel down through the common bile duct to the ampulla vater, and the sphincter of Oddi will relax, allowing the bile to be secreted into the duodenum right here. Now, bile is extremely important in the breakdown of fat because it has a quality about it, or the components of bile salts and phospholipids have a quality called amphipathy, or they are amphipathic, which means they have the special ability to interact with both lipids and water. They're both hydrophobic and hydrophilic. As you can see here, their hydrophobic side will attach to the fat itself, and the hydrophilic side will be interacting with the water or the fluid around it. And what these are going to be doing is they're going to be surrounding the fat, the large lipid droplet, and emulsifying it into even smaller droplets. And we actually have something called colipase, which is a protein attached to the outer layer, because pancreatic lipase actually has a hard time getting into the lipid. It'll attach to colipase, which will assist in the breakdown of those triglycerides. So after we have all the breakdown product, after the lipases has broken down those polymers into monomers, we have all this breakdown product, and we have, we talked about before, we have the bile salts and the phospholipids surrounding this structure, and this structure is called the micelle, or micelle, some people call it. Now these, as the micelle travels towards the brush border or the enterocytes of the small intestine, these bile salts on the outside will actually be recycled, and actually most of them, I think about 80 to 90% of these bile salts will be recycled for future use. So as it moves forward towards the enterocyte, the fatty acid or the bile salts will be recycled, and all the breakdown activity will move into the enterocyte through passive diffusion because they are lipid soluble. And they will bring along fat soluble or lipid soluble vitamins A, D, E, and K. Actually, recently we found out that cholesterol does not move through passive diffusion. It actually moves through a transport protein called NPC1L1. So when we need to absorb all of this breakdown product, we need to package all the breakdown products through this pathway, the smooth endoplasmic reticulum, the rough endoplasmic reticulum, and finally in the Golgi where it's packaged. Interestingly enough, the mechanism here is we're going to recombine the monoacetylglycerols and fatty acids. They're going to go back into triglycerides. The cholesterol is going to go back into cholesterol esters. These are all going to be packaged up by the Golgi, including those free fatty acids and the phospholipids and the vitamins. They're all going to be packaged up in the Golgi to form chylomicrons. So these chylomicrons will be absorbed through or travel through lacteals, which are specialized lymphatic capillaries that are actually big enough to take in these chylomicrons. And as you know, the lymphatic system is spread throughout the entire body, so these chylomicrons, as they travel through the lymphatic system, they can go anywhere to either the skeletal muscle for energy use, or adipose tissue for lipogenesis, or just simply back to the liver. And actually, interesting point here, these very short fatty acids, they actually won't travel through the lymphatic capillaries at all. They'll actually diffuse directly into the bloodstream. So that's how fat is digested and absorbed. So to quickly recap, we have the breakdown of polymers to monomers using lipases, lingual lipase, gastric lipase, pancreatic lipase. We have cholesterol ester hydrolase, phospholipase A2. These are all responsible for breaking down polymers to monomers. And we also have bile secreted by the liver, stored in the gallbladder, so that we can use the bile salts and phospholipids to coat the lipid, break them out emulsify them into smaller droplets. These smaller droplets are containing all of the breakdown product, and they form micelles. The bile salts are recycled back into the system, and the breakdown product will diffuse passively along with all these vitamins. They will be packaged up through this pathway, through the smooth and rough ER into the Golgi. Cholesterol actually moves through a protein, not passively. So can't forget that. And these the package product from the Golgi will form chylomicrons that will travel through the lymphatic system and will be received by the target tissue or organ or muscle. And that's fat absorption through the gastrointestinal tract. Thank you.